So today I'm going to be talking about the Nicaraguan pediatric influenza cohort. You've already heard a little bit about it, some other talks. But first I wanted to kind of highlight that I have several cohorts that I lead. The pediatric influenza cohort study is the one that we have that's been going the longest. It started in 2011. It's set in Managua, Nicaragua, and you'll hear a lot more about that in this talk. I also have a household cohort study down in Nicaragua that grew out of a transmission study. So the transmission study started in 2012, went until 2017, and then in 2017 we started the household cohort study, at first through a grant. It has now been kind of expanded, and it's supported through SEER, which Webby was talking about. And then our other big cohort study is IASO, which stands for Immunity Associated with SARS-CoV-2. So it began in 2020 with the SARS-CoV-2 pandemic. It's actually set in Michigan, so it's a domestic cohort. Everybody there is maize and blue, which are the U of M colors, because it's U of M employees, a little over 3,500 people, kind of at the height of the size of that study, and obviously to look at COVID, SARS-CoV-2. So our areas of focus across all of these studies, although some are better for one thing than another, we're really interested in correlates of protection. We've done a lot of work on that front and want to do a lot more work on that front. We're looking at the development of immunity. Here I've got a kid with a balloon, right? So in children, but for SARS-CoV-2, of course, we're also looking at that in adults as well. We look at repeat infections. So when you get flu or when you get COVID or another respiratory virus, how long are you protected for? At what level? And how much does your factors versus changes in the virus impact how long you're going to be protected for? And then we do a lot on transmission, particularly in our household study, which we have a sister study here, right, in SHIVERS-I. And there we do influenza and then SARS-CoV-2. So the INPIC study, which is the Nicaraguan Pediatric Influenza Cohort Study, is kind of the diagram is up here. It's kind of hard to point. Okay. It began in 2011. And this is the basic study design, right? We collect a yearly survey and blood sample. We provide the kids with all of their medical care through Ministry of Health health center, but by study doctors. We have an area inside of that health center, so we're very integrated with that. When the kids present with respiratory symptoms or an undifferentiated fever, we'll collect a respiratory sample. If they test positive for influenza, much like in SHIVERS-III, right, we'll collect an acute blood sample and then collect a follow-up blood sample as well. We also collect blood samples around vaccination. We collect daily symptom data when children do have flu or COVID. And then, of course, this has been going on for a number of years. So we're currently in year 12, 13, so a while anyways. So this just shows the pediatric cohort enrollment. So there are 1,666 children that are currently enrolled. 3,461 children ever enrolled. When children hit their 15th birthday, they do age out of the cohort. There are 1,513 children who've been enrolled since birth. Of those, 78% of those children are still in the cohort. So we had some kind of initial growth in aging, because at first we only enrolled children up to 11. But here you can see that the age structure of the cohort has remained relatively stable for the last 8 or 9 years. This is just showing influenza activity by year in Nicaragua. We have kind of two peak influenza seasons, really. So one kind of in June, or at least for influenza A, one in June, July, and then another one kind of in October, November, December. Typically, we usually only have one peak or the other, but occasionally we will have both. And for some reason, influenza B likes to kind of peak in between the two. So those peaks tend to occur around August. Here you can see, right, that during the SARS-CoV-2 pandemic, like most of the rest of the world, we basically had no influenza that occurred. But we had a pretty big return of influenza this year in 2022. So just to highlight a little bit about what we do in the cohort, one of the things we look at, right, I already said we were really interested in repeat infections, and because the cohort's been going for so long and we have a really high retention rate, it's a great place to look at that. We've also now had, as of like this month, almost 2,600 PCR confirmed infections in these cohort kids. So here what we did was we were looking at, if you get infected with flu, are you protected the next season or the next time that that particular type, or subtype, sorry, or lineage of flu comes about? And how does that kind of carry forward through time? So we are limited by the epidemiology, right? Because we might get H1N1 one year and then it may not roll back around for another 3 years, right? So we don't get to see each year what would have happened had H1N1 been infected, circulated. So we've got the one group, right, which was infected in year 1, and we look if that type of flu came through in the next year, what is the odds of them getting that type of flu the following year? We compare that to a group of people who were not infected in that year, in that same year 1, and look at their odds of influenza in year 2. And here you can see this is organized by the number of seasons apart. So up here we've got 2 seasons, 3 seasons, 4 seasons, 5 seasons, and then down here 7 seasons, right? Very much determined by the epidemiology. But we did see, pretty nice protection from natural infection that lasted for a number of years. So for H1N1 in particular, it was up to 4 years later. We're still seeing really significant protection. And in fact, at that kind of, 2 to 3 year mark, so down here you can see this is a 0.1, right? So at times, you're talking 80 to 90% protection from reinfection over a 2-year time period, right? Comparing that to the vaccine, right, where we get often less than 50% vaccine effectiveness or even much lower than that. And usually it does not, always it does not last even to the next season. So this is just to kind of show you, we've done that, not only for H1N1, H3N2, B Victoria, and then B Yama, we had fewer seasons of B Yama. But this was fairly consistent across the different flu types where we were seeing significant protection for a number of seasons. The protection was kind of the shortest for H3N2. Not really surprising because that's the virus that was changing the most, at least over this time period. And I'm not going to show you it here, but we actually used information about how much viral change was happening to better understand, too, when we saw deviations in the pattern for how long individuals or kids here were protected. And so this is just kind of showing the seasonality of, I guess they're a little hard to see, of influenza kind of prior to the pandemic. And then here in the pink colors, we've got 2020, 2021, so the first 2 years of the pandemic, and then this year, 2022. And much like here, we saw very early or off-season influenza circulation, right? So we actually saw peaks of influenza in months when over the past 10 plus years, we've never seen any flu down in Nicaragua. So I was asked to comment on some areas for synergy or collaboration. I think there are a ton of really obvious things, a lot of things that we can collaborate on. But I think it'd be really interesting to think about how we can do a comparison of influenza in the northern, southern, and tropical regions. One of the things that infectious diseases is not particularly well known for is replication or validation, right? You see one thing in one population at one time and you're like, it's a fact, we're done, we're moving on. So I think, the fact that our cohorts are so similar in their designs really presents us with a very unique opportunity to show that across multiple populations and make sure it's not just something that was happening in one particular location at one time, but actually a biological fact. Also doing it across multiple seasons is really valuable for that reason as well. And then, of course, there's the opportunity to combine samples and data from SHIVERS, Los Angeles, and then the Nicaraguan cohorts. Paul showed those things of like the series of exposures. And one of the things we really want to look at in these cohorts is imprinting. And people can get a lot of different sequences of exposures, right? So even when you have thousands of kids, you end up getting very small groups when you're looking at third or fourth exposures if you want a particular series of exposures occurring. So I think that's another really good area where we can think about how to figure out good projects to collaborate on. And with that, I'm going to say thank you to the team. So we have a huge team. Actually, you need to get some more updated photos of the Nicaraguan team. These are all kind of pre-pandemic, but we have about 120 people that work down in Nicaragua on these studies, particularly Angel Balmaceda, who's the head of the National Virology Lab down there, and Guillermina Kwan, who heads up the health center where we work out of and really heads up the field work. And then this is my team in Michigan, who do a ton to analyze all the data, clean all the data, and then support all of these studies. Thank you.